This system can be used to expose engineered hard tissues to varying regimens of afterload in order to study the effects of these stimuli on tissue force development, remodeling, and maturation. Our technique allows one to precisely subject beating tissues to a broad range of customizable afterload routines over prolonged periods of culture without even needing to open the cell culture plate.
Our method could be modified to control afterload in other muscle tissue culture systems, such as skeletal muscle, smooth muscle, or excised papillary muscle.
Since our systems approach is rather unique and features technical aspects that many scientists are not familiar with, visual demonstration may aid in recreating our setup.
To manufacture the magnetically responsive silicone racks, acquire the 24-well plate-compatible racks of silicone posts described in the text protocol. Keeping a fixed polarity, lubricate the magnets with water and insert them one at a time into the outermost posts of the silicone racks. Use a blunted piece of stainless steel dental wire to carefully push them to the bottom of the hollow post cavity. Up to 5 magnets can be stacked in each post. Use round-nose pliers to bend stainless steel dental wire into braces 11.25 millimeters wide and 15 millimeters long. To ensure the correct dimensions are achieved, one can use a self-made jig to aid in the wire bending. Then use wire cutters to cut the braces and a file to smooth the cutting surface. Lubricate the braces or posts with water and insert them into the silicone rack, fixing the second and third to the outermost post in the process. Begin preparing the afterload tuning device.